Couple things we have to do is, for some reason, I neglected to tell you when your exams are. I noticed that I made note of that the other day when I was like, oh, they should be having an exam coming up soon. And then it's like, oh, I said to myself, we figured out that we were going to do 4 exams, right? And then I said, I haven't told them when their exams are. So, so you know, okay? Exam 1 will be on February 21st, okay? And I did this so that it's not at the same time as chemistry, okay? So February 21st, exam 1. Exam 2 would be on March 28th. Exam 3 is on April 18th. Okay? And then your final. Your final will not be cumulative. April 18th. Okay? So exam 1, 2, and 3. Alright? 21st, the 28th, and the 18th. And then your final would be probably 2.5 weeks after that last exam. Okay? There's the material for the second exam. Okay, we'll probably stop before spring break, but we'll just have the exam later. All right, so that the last 2 exams, or exam 2 and exam 3, where we stop the material for the exam will be a little off because of the timing.
The bio exam is May 8th.
Okay, so your final exam is scheduled for May 8th. Look at that, 8. All right, isn't that nice? Okay? Questions, comments on exams, when they are. If there's problems, I'll deal with those on an individual basis. All right. So, the next order of business is I want to go over something that we sort of rushed through on last Friday. Okay? And the reason why is we didn't cover it the way I'd like to. Right? And so we went through DNA replication, and then we sort of rushed through DNA packaging. All right? And how DNA is packaged in a cell. All right? And I want to make sure that you go through this because it's important in understanding sort of how DNA is accessible. Okay? So if I have a double-stranded DNA in a linear sequence, all right, that if I pulled it out all the way and removed all the twists, it would be about 6 feet in length, right? But only about 2 nanometers in diameter, right, from one sugar phosphate backbone to the other. Okay? So there are certain levels of packaging that DNA goes through, or that DNA is subjected to, in order to fit that 6-foot linear piece of DNA, which is roughly about fingertip to fingertip, okay, into a cell or a cell nucleus, which is about 10 to 50 microns in diameter. Okay? So there's a lot that has to go on in between them. All right? So if you take that 6-foot piece, the first order of DNA packaging is the double helix itself. Okay? So the first order packaging is putting it into a double helix. Okay? Because of the helix itself, that 2-meter piece of DNA twists and then compresses that overall length, down. Okay? From there, now this, I'm just going to draw this as a single strand, all right? The next order of packaging involves what? What did we say? Nucleosomes, okay? And nucleosomes are a complex of DNA and proteins. All right, so we're gonna talk about nucleosomes, and they are DNA and proteins. All right, those proteins are called histones. All right, and every nucleosome has 2 copies of 4 different histone proteins. All right, so really, a nucleosome is what we call an octamer. All right, it has 8 different subunits. Okay? And so if I was to draw a histone, it looks something like this. All right, and then it would have another set of proteins behind it. All right, a little spider looking like this. Okay? So this, what you see in circles up there represents the core of the histone protein, and then you have extensions that go beyond it. All right, those are called histone tails. All right, the tails are important for accessibility. All right, you will, when we get to transcriptional regulation, which we're going to do next, we're going to talk again about these histone tails. Okay? Because what's going to happen now to the DNA in packaging around this, in forming a nucleosome? The DNA is going to wrap around the histone. So here's our DNA, and it's actually going to wrap around the histone like that. Okay? And come out the backside. Okay? So it wraps around. And when it wraps around, you have exactly 146 base pairs that interact with the histone. Okay? So the DNA is wrapped around. The tails are important for those histones being modified and will release the DNA when necessary. All right? Because we know for replication what has to happen. DNA has to unwind, right? And if DNA is bound to proteins, they get in the way. All right? They get in the way of the normal replication machinery, all those proteins that are important for replicating DNA. Okay? How do you think researchers found out that 146 base pairs was wrapped around DNA? And there's exactly, another 50 base pairs between this nucleosome and the next nucleosome. What's that? Not to the diameter. So what are you doing in lab this week? Gel electrophoresis, which we talked about on Monday, right? That separates different pieces of DNA based upon size. Okay, so how do you break down the DNA into different pieces? What paper exercise were you doing? What was that showing you?
The plasmid.
Plasmid being what?
Transformed.
Being broken down and cut by enzymes, right? So those enzymes are nucleases. They recognize DNA and they'll break it up. Okay? But those nucleases depend upon DNA being what we call naked, not bound to proteins. Plasmid DNA is not bound to proteins. Okay? So what researchers did was they found a way to isolate this level of structure. Okay? They took an artificial piece of mammalian plasmid, mammalian DNA, and incubated it with nucleosomes. Okay? And spontaneously, they'll incubate it with histone proteins, and spontaneously, they will form this structure. All right? Now, when you take a random enzyme, or an enzyme that breaks down DNA called DNA ACE, all right? Where is that DNA, where is this protein going to be able to break DNA down into nucleotides? In between. All right? So it'll break down here and here, but it won't break down the area or the DNA that's bound by the nucleosome. Right? So then researchers stopped the action of the nuclease and then isolated the DNA, and then they separated it all out on gel electrophoresis. And they found that they always had this sort of band at 146 base pairs. All right, so that's the way they found sort of the structure of or how much DNA wraps around a nucleosome. Okay? So let's go on. So now we have DNA, all right? That is, and we have a bunch of nucleosomes, and we have DNA that is wrapped around nucleosomes, right, giving you your sort of beads on a string structure. Okay? From this point forward now, it's a matter of placing nucleosomes in a certain area so that they have enough space. All right? So that this is what's called a 10 nanometer fiber. It's a 10 nanometer fiber. All right? The next order of structure is this 10 nanometer fiber forms a 30 nanometer fiber. So 10 nanometer fiber meaning the diameter of from one side of the nucleosome to the other. Okay? So in order to make a 30 nanometer fiber, what do you have to do? You have to place those nucleosomes sort of next to one another so that they are 3 nucleosomes across. All right? We had a question, Sarah?
I was just wondering what the 31 looked like.
So yeah, we're going to so here's nucleosome 1. Okay? And I'm not going to draw the DNA around it yet. All right? So if this is nucleosome 1, the next nucleosome is packaged in an area adjacent to it. Okay? And then nucleosome 3 is packaged in another area. Okay? So that they are sort of arranged in a certain way. Okay? In space. Think about 3-dimensional space, one above, slightly below, a third, sort of off to the side and slightly below. Okay? Then you have, I'll draw the DNA in here now. So the DNA is wrapped around there, wrapped around there, wrapped around here, and then it has to go to the next one, which would be behind it. Right? So then you have your next nucleosome, and so on. So that the nucleosome are packaged in space, right? The histones are packaged in space so that they are separate from one another. What it causes then is another winding and a diameter of 30 nanometers across. Okay? So then that looks like, we'll say that then looks like this black line. Okay? Now the next step is going from a 30 nanometer fiber to a chromosome. Okay? And so this black line, or this 30 nanometer fiber, then loops. Like that. So that you have these two sort of very close looking loops. Have you ever seen, actually, you guys have played with pipe cleaners before? right? The little fuzzy things. If you imagine that the metal piece of the pipe cleaner is the proteins that are within this portion that sort of comprise the chromosome that DNA loops around, and then the furry stuff is the DNA itself, right? That's sort of what it looks like. All right? And that ends up making up your chromosomes that look like, that we draw like this. Okay? So that this part looks like that, and then this little part right there looks like this, and then one of these looks like that, and then goes down. Okay, so hopefully that better explains the structure of chromatin. Okay? This is how DNA is packaged inside a cell. All right? And most of the time it's sort of in this structure, okay, in terms of chromatin, and then it goes back and forth between this and chromosomes for replication, for mitosis. All right? Questions on the packaging of DNA? All right? And then we have the two forms of packaging. We talk about heterochromatin versus euchromatin. Heterochromatin is very packaged, lots of protein. It is not transcriptionally active, right? Because of all the proteins that are there, it's binding DNA. It prevents other proteins from getting in. Okay, it's not, the DNA strands aren't accessible to proteins.
Say that again. What's not transcriptionally?
Heterochromatin.
Okay.
It's not transcriptionally active, right? It's sort of shut down and compressed, compacted. While euchromatin, okay, EU chromatin, that is transcriptionally active. It's less packaged, there's less proteins there, the packaging can come and go, it's open, all right, it is transcriptionally active. Okay, questions on heterochromatin versus euchromatin and what it means? Sarah?
Are they just like one chromosome is like randomly heterochromatin, or are certain ones found in like different?
Well, let's use a great example, okay? Let's see, we have probably 70 to 80% women in here. That's probably 70%. Women in here. What happens to your X chromosomes in all your cells? Are one of your X chromosomes produced or expressed and the other not expressed? Right? We talked about this last semester, something called X chromosome inactivation. Okay? So randomly, at some point during development, a cell will decide to inactivate an X chromosome. Okay? That inactive X chromosome is packaged in a heterochromatin structure. Okay? It doesn't go away. That DNA doesn't get, because when a cell replicates, that has to come back. Okay? But it is in a form, an inactive form, so that you don't have double the amount of genes on the X chromosome being expressed as necessary. Okay? Is that a good example of heterochromatin? Catherine?
Do men have heterochromatin?
Yes. Yes. All organisms have heterochromatin. Okay? Then brings us to chapter 14. All right, so I wanted to revisit the structure of DNA and how it's packaged, because this is important in terms of regulation of transcription and translation. Okay? So in chapter 14, we'll go over transcription, which is what? Knocking stuff over. Which transcription? DNA to RNA. So we're going to take a gene that's in DNA, and we're going to make an RNA transcript. Okay, we're going to transcribe the DNA code so that it can make a protein. Okay? And then, so then we go from transcription and then to translation. Translation is then transferring that code that's in RNA to a functional protein. Okay? And so we're really relaying information. The information that's found in genes, right? The order of amino acids, what the function of that protein is. Okay? And we're relaying that information from the nucleus to the cytoplasm and producing a gene. This is globally what's called gene expression. All right? So it's the process by which DNA directs protein synthesis. Your two stages, transcription is making the RNA transcript, okay, decoding the code, all right, and then translation is putting that code into a functional protein. Okay? That's sort of the overview, what we're going to look at. Within that, we're also going to talk about the steps of transcription, how it's regulated, all right, the different levels of regulation, all right, what's important for, we're going to compare and contrast bacterial transcription and translation to what happens in eukaryotes, and you will understand the entire process of transcription and translation and what's important for mammalian cells versus bacterial cells and so on. Okay? So there's lots of ins and outs in this. All right, but first, how was the, I want to go through how this was thought up, all right? And how the idea of, prior to this, we know that, all right, here we have this trait. We'll go back to Mendel. Traits are passed on, okay? And then, traits are found in DNA. And then we figured out that every trait is associated with a gene, all right? But now the idea is, well, if genes are made up of DNA and protein, is DNA have the code? How does DNA translate over to a functional protein? That's sort of the logical progression of understanding. All right. So how is the relationship between genes and proteins discovered? All right. In 1902, British physician first suggested that genes dictate phenotypes. All right, and this was just sort of an idea based upon observation. All right, genes dictate phenotypes, and he noticed that there were lots of inherited metabolic diseases which were disruptions in enzymes. And enzymes, all enzymes are what? All enzymes are proteins. So the idea was something that's inherited, passed on from one generation to the next. We already discovered that was, through the work of Mendel, etc., that it was genes. Something that's inherited from one generation to the next leads to a problem with protein, with enzymes. So the inference was one gene, one protein. Something wrong in the gene leads to something wrong in the protein. All right? So that's his observation. All right? Also with that was cells synthesize and degrade molecules in a series of steps called the metabolic pathway. All right? These are your enzymes that he was talking about. All right? So cells synthesize and degrade molecules in a metabolic pathway. So you have, say, substrate A that goes to product B and then to substance C. All right, say this is the production of testosterone or estrogen. Okay? This is the steps that it takes. All right, it starts with cholesterol. An enzyme catalyzes the production from cholesterol to, say, 19-nortestosterone, and then another enzyme catalyzes the production from 19-nortestosterone to estrogen. Okay? So you have enzyme 1 and enzyme 2. All right? This is a metabolic pathway. A substrate is broken down or changed from one to another. And then so then that product is the substrate, right? Serves as the substrate in the subsequent reaction. But a change or disruption in the gene that codes for enzyme 1 would do what? Alter the process. Okay? So if I have a cell that I disrupt the gene for enzyme 1, what's going to happen to the production of product C? Nothing? Anything? Something? What? Bad things. Thanks, Nick. What's going to happen to the production of product C? Okay. It won't be produced. Okay. If I produce, what's going to happen to the production of product B if I have a disruption in enzyme 1? It won't be produced. If I have normal enzyme 1, but I have deleted enzyme 2, what's going to happen to the production of product C? It won't happen. What's going to happen to the production of product B? It'll be there, okay, or it'll be utilized, okay? It will be produced, all right? If I have a disruption in enzyme 2, but not enzyme 1, what's going to happen to the levels of substrate A? They're going to decrease, all right? And then you'll have an increase in, or a collection in excess of product B because it's not going to be utilized in the next step, all right? And researchers use this to their advantage, all right? All right? And you'll see that in a second. So, I'm going to add to existing works. So what they did was they disabled genes in a bread mold. Okay? So they took this mold that grows on bread, they exposed it to X-ray radiation, which you know from your last chapter causes disruptions in DNA structure, causes mutations. All right? And so they study this bread mold, and because it's easy to detect these recessive mutations, and they study the mutations that alter the ability of fungus to grow on minimal media. Okay? Now, this is important for their process. All right? Minimal media meaning giving them the minimal amount of nutrients that they need. All right? So that they have everything that they need within themselves, in their cells, to grow. All right? So that means growing on minimal media requires all their enzymes and such to be normal. Mutations in that will disrupt their growth on the media. Okay, do you guys understand that? Kind of? Okay. Next. No. So here you go. Here's your neurospora cells, your bread mold. They expose it to X-rays, and then each surviving cell that is, can grow on the minimal media now forms a colony that is just really clones of the original surviving cell. All right, so they're all the same identical cells. They isolate those colonies, and then they next step. Okay, so here's your control, your non-irradiated cells. Okay, the wild type cells grow in minimal media, like you expect. The surviving cells are tested for inability to grow on minimal media. All right? There's no growth. What does that tell you? So what happened? What caused that no growth? There's a mutation somewhere that's causing them to not grow, there's a disruption in a metabolic pathway somewhere. And somehow they're not getting one of their required nutrients. All right? The next step then is to sort of add back. Let's start to add back to the cells that aren't growing to see what they need in what they're missing. Okay, what can't they produce? That will help them grow. So the mutant cells are placed in a series of vials, each containing minimum media plus one additional nutrient, right? This is a nutrient that's sort of along that metabolic pathway. So we go back to our ABC analogy, right? A to B to C, okay? If the cell requires C to grow, right? And enzyme 1, enzyme 2, all right? If the cell requires C to grow and you mutate enzyme 1, is the cell going to grow? No. Okay, why? Because let's say you're only giving it A. A is the only thing that's available to it. But the cell will break down A to B to C and produce the minimal nutrients. Okay? So if you have a breakdown in enzyme 1 and the cell needs C to grow, is it going to grow? No. Okay? But now you add back B. Okay? Is the cell going to grow? Yes. Why?
Because it's the in between A and C.
Right? Because B to C, it requires C to grow. That step is reconstituted. See what I'm saying? So if you add the nutrients before it and that enzyme is available, it will produce the necessary nutrients. Okay? If you have a breakdown in enzyme 2, okay, what's going to happen? There's going to be accumulation of product B, right? But there still won't be any growth because the cell requires product C, right? You add product C, cells grow. So that's sort of the idea here. Let's add back and see where there's a disruption along the pathway. All right? So they amassed a collection of these strains and catalog the results, right? Example, one set of mutants all required arginine for growth. Okay, arginine's an amino acid, important for growth, right? And we're going to go through this in a second. Different classes of these mutants were blocked at different steps in the biochemical pathway for arginine synthesis. All right? This is your A to B to C sort of progression. All right? So here's gene A. Here's your precursor, right? Eventually, cell requires arginine. They all need product C, right? They all need arginine for growth. The process of producing arginine is done by cells, okay? Given the original precursor. The cells will break down whatever substrate in a metabolic pathway to produce arginine so that they can produce proteins for growth. You disrupt any of these genes, A, B, or C, you stop arginine growth. Okay? So if strain 1, okay, disrupted gene A, what would you have to give to it in order for the cell to produce arginine? Ornithine, right? You have to provide the next substrate. So you disrupt gene 1, that strain will not produce, or you disrupt gene A, excuse me, that cell will not produce or catalyze the reaction from precursor to ornithine. So you add ornithine, cells grow because now that precursor, the ornithine's the substrate for the next subsequent reactions. Okay? So this led to the one gene, one enzyme hypothesis. All right? So the question is, you have to add the substrates and not the enzyme. You could add the enzyme, okay? But the idea was for the researchers were, well, if there's a definitive break in gene A, okay, I could add the enzyme, but does that really prove that that doesn't do anything? Or that the bread mold will grow given that the other enzymes aren't disrupted, right? So if you had, say, gene A and gene B, if I added gene A, what's that going to do?
It's not gonna get to.
Is there going to be growth? No. So the idea was, let's go back, add the substrates along the pathway and see where definitively there's a disruption. So if in this case, you could have two mutations, right? If I added the precursor and ornithine, is there going to be growth? No. But if I add citrulline, there would be. So what does that tell you? That there's a substrate that above this, there's a disruption above the citrulline and above the ornithine that are both, there's at least two mutations. So it was the way, instead of adding the enzyme, they could just add the substrate and let the cells do the work to definitively show where along the pathway mutations were.
It was to show the metabolic pathway more than anything.
Yep. So it was two things, that there was disruptions in the genes and that it was within this metabolic pathway. All right, other questions? All right, we'll stop there. But you know that some enzymes, all enzymes are proteins, yes? Are all proteins enzymes? Well, thank you, Alyssa. Right? Not all proteins are enzymes. You have structural ones, you have antibodies, you have other stuff. All right? So, at first it was one gene yields one enzyme. Well, then you realize that there's more functions to proteins than just enzymes. So now it went from one gene, one enzyme to one gene, one protein. All right, and it was like that for a while. All right, but then through lots of DNA techniques, we realized that many genes code for many peptides or different parts. All right, example, the beta globin gene, right, which is important for the production of hemoglobin. All right, can actually, depending upon how the transcription occurs, can code for several different polypeptides. All right, based upon structure. All right, so then it went from one gene, one protein to really it's really one gene, one peptide. Right? And that's sort of where it's at this now. All right? Or one gene, one polypeptide. For our purposes, protein and polypeptide are basically synonymous. What's the difference between a gene and a polypeptide? I'm sorry, what's the gene between a protein and a polypeptide? Cat. Okay, it's not a developed protein. What is the defining difference between the two? Functionality. Okay? So a protein is functional. It does its job biologically. All right? Hemoglobin binds oxygen. Okay. That is a protein. The subunits of hemoglobin, all right, are polypeptides. They are not together as a functional unit yet. Okay, so functionality. Questions on sort of the determination of those? Sarah?
When you had the picture that had the different proteins for the bread mold,
Mm-hmm.
why isn't it that the enzyme isn't? You said you needed to add the B, so why is it that you don't need to add the enzyme?